As I mentioned, I'm Phil Rooney, and I joined Smithers as a Director of Chemistry here in the UK earlier this year. I worked previously within contract research for many years and spanning both technical and operational management roles. My background includes time as a GLP study director, working within disciplines of environmental fate and metabolism. I've also held operational management roles, leading teams through delivery of GLP studies to support environmental risk assessments and regulatory submission. Okay, so that's me. Moving on, a little bit about Smithers. Um, at Smithers, we partner with our customers to generate high quality data that supports sponsors and regulators to understand how substances impact the food chain and the environment. We do this through a wide range of GLP compliance studies covering expertise within residue chemistry, substance dissipation, metabolic fate, in a wide range of environmental compartments. We also look at bioaccumulation, ecotoxicology, and phys-chem characterization. Laboratories are based in the UK and the US, and we run studies under OECD and EPA guidelines. We support higher tier, more bespoke investigations when standard approaches don't quite fit. So, moving on to some introductions for the speakers of today's session. Joining me today are two of our experienced colleagues within the field of environmental chemistry, Mitch Kelly and Sydney Blooming. I'd like to just take a moment just to introduce both. Let's start with Mitch. Mitch joined Smithers in 2015, and he brought with him an incredible depth of experience, 38 years in analytical chemistry, working across both contract research organisations and governmental testing laboratories. Mitch has built a strong reputation as a go-to technical expert within our team, especially when it comes to method development, analytical troubleshooting, and looking at compound behavior in complex test systems. Hi, Mitch. Thank you for being
Hi.
here today. Sid has been part of the team for 20 years and has experience in a wide range of analytical disciplines, particularly in the field of residue chemistry. Sid's role it centres around the development and application of methods to detect residues in soils, agricultural products, biological tissues and fluids. So welcome, Sid. Thank you for your time today. Okay, so moving on. I just want to frame today's topic. So I'd like to start with a quick poll, just to get a feel for the experience within the audience today. So there's a particular piece of guidance that's relevant when dealing with challenging substances, and that is OECD 23, which was finalised in 2019. And basically, this guidance document has a range of methods and applications that you can use to look at difficult to test substances. So if you could just take the time to answer one of the questions, how familiar you are with this particular guidance document, if you have a deep understanding, if you've heard of the guidance document before, whether you've never heard of it before and would like us to align you, then please fill out the choices. Okay, great. So the results are in. Right, so looks like we've got a real diverse set of experience within the audience, so that's great. So hopefully today's session will be some benefit and provide you with a bit of more knowledge on that particular guidance document. Okay, so moving on, I will now pass you over to Sid. So thank you, Sid. Please take it away.
Thank you, Phil, and thank you everybody for joining us today. Quick introduction. So we'll just go through quickly what we're going to go over today with myself and Mitch. The first few slides I'll go over. We'll start with what makes a substance difficult to test, what specifically makes a substance difficult to analyze, what do we need to achieve with OECD 23 in aquatic ecotox testing. We'll go through a few examples. Why do traditional methods fall short with some difficult substances? Then we'll pass on to Mitch, where we'll go over practical approaches, this sort of essential toolbox of equipment that we use, some advanced solutions, how do we stay ahead? We'll conclude, and then we'll do a Q&A session. Right. So according to OECD 23, if you actually read the document, it does list what sort of substances we consider difficult to test. And this is very much in the context of aquatic ecotox testing, so chemicals that we will analyze from media that we use in ecotox testing. And according to the document, ones we the chemists would like to avoid, if possible, is poor water solubility. Obviously, in an aquatic test system, if it's poorly soluble, it introduces difficulties. Toxicity, low concentrations. What that means is that often you then have to test a very low limit of quantitation. That adds difficulty. Uh, Volatile, it evaporates easily. Light sensitive, so it degrades in light. Hydrolytically unstable is something we don't like either, because it just reacts with the water and it transforms into other products. Oxidizable as it reacts with oxygen, could be subject to corrosion or some sort of transformation. Forms colloids, so for perhaps insoluble substances, they might form sort of suspensions that we call colloids. They can be biodegradable, so not the test system organisms might help degrade them, but also local organisms in your test system. Something that's adsorbing, so it sticks to all sorts of surfaces. That can make difficulties. Is it complex with, say, metal complexing? Is it colored? Does it attenuate light? Is it hydrophobic? That again ties in with solubility. Is it ionizable? This makes it very soluble, for instance. Again, there is some overlap there. Is it a multi-component substance like a UVCB, for instance? You have different substances in there. They all might have different properties, and that gives us difficulties. Or it's something with surfactant properties, like you see in detergent, where it then becomes very hard to pin down the actual solubility of the substance. Okay, so in an aquatic ecotox test, what are they trying to achieve with test organisms? When we actually dose the water with our test substance, they want to obviously achieve target concentration because this aids the risk assessment. You want to be able to maintain that target concentration during the test. When required, you might want to achieve water saturation if it's a particularly difficult to dissolve substance. If there are products, you want to be able to understand the toxicity of those products. You have to be able to maintain nutrients for the system, define those exposure concentrations. You want to be able to prepare representative solutions, and if necessary, you want to characterize the chemicals. But our focus today is on the chemistry side, where analysis, we want to analyze and quantify the exposure concentration in aqueous media, and we want to do it accurately and precisely. Then we know that if we do that, well, then we know that the organisms have been exposed to the amount of substance that we want it to. That's our focus for today. Thank you. So, but that's OECD 23. To an analytical chemist, what specifically makes a substance difficult to analyze? The first one is stability. An unstable substance often presents greater logistical challenges for us. It can increase time. It can have cost implications. Some examples might be that it absorbs light between those wavelengths, or it's volatile. That can form part of what stability is. Some of our mitigations can mean speed of analysis. You just have to do it quite quickly. You might need special storage conditions, special handling conditions, and use of stabilizers where possible. It's worth mentioning as well that it's not just instability. It's a stable substance that has absorbing properties can also cause problems for an analytical chemist, because they can cause especially contamination problems in the whole system. As I say, with absorbing, that can cause difficulties. Usually as a general rule, it's anything about with a log PaW of greater than 4. And these can stick to the glass, plastic, injection systems. During the analysis, it can cause cross-contamination, or it can cause underestimation of the amount of substance present. Also highly ionizable substances. Very similar to polar substances, they one of the problems they cause is, at least in conventional methods, is they are poorly retained on conventional LC systems, and this can cause problems. We'll go over as we go along. Okay, so the next two are there's a lot of overlap between them, but what makes it difficult is highly polar or highly non-polar substances. The two extremes. Highly polar substances, they retain poorly on standard liquid chromatography columns, and that results in poor separation from other matrix components. So you get matrix interferences. You can get highly non-polar substances. These are poorly soluble in aqueous solutions, and they often don't readily ionize in analytical systems. And this is something that can cause difficulty, especially if we are analyzing by mass spectrometry. Then, of course, highly soluble and insoluble substances. These really overlap a lot with highly polar and non-polar. They often share a lot of properties, but in general rule, we're looking for a solubility anywhere less than 100 mg per liter would be classed as insoluble, or at least partly insoluble. As I said, there's overlap, and again, highly soluble, we get poor chromatographic performance, lack of retention, and again, highly insoluble, poor aqueous solubility, and does not readily ionize. There's a lot of overlap there. Okay, so we'll go through some examples of difficult substances that we could get. So the first we have PFAS. As I'm sure most of us have heard of these forever chemicals that are appearing on the news a lot. An example would be perfluorooctane sulfonic acid. Excuse me. And the other example is TFA. This was a late addition to our presentation, trifluoroacetic acid. Some people might have heard of it as something we often use as a mobile phase additive in chromatography, but it's also been on the news lately. If you're in the UK, on the BBC, about that they've recently found unacceptably high levels of TFA in United Kingdom river water. So this is something that even made the news on the BBC. So it's certainly something that's gonna come up more often. And so the kind of properties we look for in PFAS, to be honest, it's quite a wide variety of properties. Anywhere from low volatility, does not easily ionize, it can be extremely stable and ubiquitous in the environment. Some of the challenges can include instrument amenability. Very low limits of detections are needed for lots of assays, and it often requires derivatization. This all carries challenges. We also have plasticizers and flame retardants. Example being trifenophosphate. They're persistent, they're very insoluble, and they're ubiquitous in the environment. And this can cause difficulties in cross-contamination problems. Difficult to find a matrix that's actually free of it that we can test on. Silane, siloxane, and silanols, example being triethoxysilane. So in water, they're quite unstable. They hydrolyze, they form condensates, oligomers, polymers. It makes it difficult to track exactly what's going on in the system. We got multiple degradation products. These products can be very polar and can be challenging to chromatograph. Then we have detergents, surfactants, and dispersants. Sodium lauryl sulfate is an example. They can be unstable, varying characteristics, form micelles, complex structures. Main implication is the solubility can be quite challenging to assess, and this has special implications for aquatic ecotox testing. Thank you. So we just go on to the last few. Getting a lot more attention lately is biopesticides. Certainly environmental friendly, but can offer challenges to the chemist. We might be talking about natural pyrethrins, RNA, neem oil, especially with gets some attention with its insecticide properties. They can be very, very unstable, multiple components. Again, difficult to quantify. Might require uncommon techniques. When I say uncommon, at least to conventional environmental chemistry labs, we might be talking about sort of instruments like ELISA and PCR. and electrophoresis. We've got biopolymers as well. These are sort of biodegradable plastics or cellulose or starch. They can range from unstable, high molecular weight, very complex mixtures, insoluble and organic solvents. Again, just difficult to quantify, lack of uniform repeating units, and they can degrade and just generally complex chromatography and similarities with industrial polymers, plastics, synthetic rubbers, organosilicones, large molecular weight distribution, branching, cross-linking. Again, very, very complex chromatography, and just varied reactivity, and can offer a lot of challenges. Those problems I went over, they're certainly not insurmountable, but they definitely can make life a bit more difficult to the chemist. And but one thing I just wanted to take a sidestep on this one slide. We're talking about difficult, challenging substances, but we shouldn't forget that there are other aspects to a method that can offer challenges, and we'll just quickly go through that. And I just sort of call it the little trifecta of difficulty. That if you're dealing with a difficult analyte, and perhaps you have a challenging limit of quantitation, and then your sample becomes complex, it just compounds the difficulty. And with these, and some of these can include complex matrices. Water generally isn't a complex matrix, but it can become complex when it's been aged and there's a lot of algae growth and a lot of organisms excreting into it. You have the challenge of a low limit of quantitation that makes it more difficult. In other examples, scarcity of matrix might cause difficulties. For novel chemicals, availability of standards can be a challenge. Are these materials hazardous, difficult to handle, difficult to process? And there's obviously a question about instrument performance and amenability. So finally, before I pass you on to Mitch, I'll go over why traditional methods fall short or can fall short. What I mean by traditional is these sort of older, more iterative methods that we've traditionally dealt with, where we didn't quite have the instrument performance available, so the methods often had to have a lot more workup, where they had to be concentrated and cleaned up more. And you see there in the diagram the kind of typical steps we have to go through when our methods tend to be more iterative. So that's what I mean by traditional methods. And those traditional methods, they can be multiple step, time consuming, and for an unstable substance, they can give them time to degrade, and that just makes it difficult to quantify. Our traditional methods, they can fall short because nowadays we might have more controlled, efficient, and targeted use of traditional methods. They might require lower levels of detections than traditional methods can deliver, such as when I say targeted, more efficient use of pesticides, for instance, there can be drone technology and the use of AI, and because of that, we need lower limits of quantitation. We might also have to deal with novel chemicals designed not to accumulate in the environment. So again, we're dealing with low limits of quantitation. Generally, traditional methods, they reverse phase and they rely on substances being hydrophobic, whereas the more novel, environmentally friendly chemicals can be highly polar, and that can add difficulties, but that's certainly not a bad thing for the environment. And sort of traditional methods are often designed for small molecules, while maybe with biopesticides or biopolymers, we might actually require a different approach. We might have much larger molecules to deal with. It's sort of your traditional environmental lab. We might not have the experience set for that. And you have to remember there may be structural and chemical similarities between environmentally friendly substances and endogenous substances. So it can be difficult to actually distinguish them. So I just want to thank you for your attention. At this point, I'm going to pass it over to Mitch. So on to you, Mitch.
Thank you, Sid. So we'll start to look at some of the practical approaches to dealing with these problems. So first up, we'll think about phthalates and pyrethroids. These are hydrophobic. They're highly absorptive. The log Ps for these are 6+ usually. So The other thing is that many of these, they're quite ubiquitous. You end up entraining them into materials from packaging, even from gloves. So you've gone ahead and you've gone out and you've sourced high quality consumables for use in your methods. And then you find you've actually got to go in and do some further cleaning of things like vials, pipettes, sample tubes. So you're looking at perhaps acid rinsing, solvent rinsing, or the old favorite with something like phthalates and pyrethroids is simply to put the whole lot into a laboratory furnace and use temperature to degrade what's on there. And then you've got a clean substrate to start working from. Your reagents, your solvents that you've spent a lot of money on sourcing high quality ones, you then go ahead and actually carry out a screening analysis and make sure that the background concentrations of what you're looking for is sufficiently low that they don't interfere with the quantitation of your target compounds. And once you've done that and you've identified that batch of solvent, you then squirrel that away somewhere and keep it away from everyone else's use until you've finished. Because otherwise, if you get into a new batch of solvent, you've got to go through the whole process again, and you're not guaranteed to find that the next one's as clean as the previous one. You have to segregate work very carefully and think about situations whereby, for example, with pyrethroids, somebody in the lab might be doing a 5-batch analysis on a pyrethroid formulation, and they've got tens of % of active ingredient in their large pot of field use insecticide. And on the other side of the lab, you're trying to measure 1% of the saturation concentration in aqueous media. That's a situation you want to avoid very carefully because the risks from cross-contamination are A, very real, and B, very significant. And again, that alludes to the next point, the understanding those routes of contamination. Production of gloves and even packing laboratory gloves. Everyone thinks wearing gloves is a good idea, and generally is, but gloves go into boxes which are printed and have various other chemicals used in their manufacture. They transfer onto the gloves and then have the risk of transferring into your samples. An interesting one we've had is the use of personal care products. No one would argue that it's good advice to wash one's hands frequently and regularly when you're doing more or less anything, and lab work in particular. But if you're looking for something like detergents, then your hand wash, which is over every sink in the corner of every laboratory, is a massive potential source of contamination. Moving on to other plasticizers and fire retardants. Again, you've got things with very, very low water solubility, highly lipophilic. Here you come up against measurement of true solubility. In many cases, you've got values given for water solubility which are less than mg per kilogram, mg per liter, something like that. Those are usually artifacts from the times of less sensitive methods. In terms of trying to do a modern risk assessment or carry out a more nuanced test, that value's not useful. So having to go back in and sort of carry out phys-chem tests again. True solubility is an interesting one. Most of the time, if you're going to make a solution and decide, yes, that material's in solution, you might use something like Tyndall effect or critical micelle concentration to assess whether what you have is actually a true solution or some kind of microdispersion. The problem with materials like these is that the concentrations you're looking at are way below where Tyndall effect or CMC is actually going to give you a value. And that's critical to understand, because if you are way below the saturation concentration, there's a risk that you're not testing at the maximum water solubility and that the data you produce is actually a false negative. But on the other hand, you cannot be above the saturation limit. The guidelines are very clear that you must not exceed the saturation concentration. So with that, and then understanding your sources of background contamination, where they come from, and controlling for those. As could have the next, thank you. So an example among pyrethroids is cypermethrin. Cypermethrin is, as all pyrethroids, it's an insecticide, it's a semi-synthetic pyrethroid. It's widely used in agriculture, in animal husbandry. It's also used in public health, things like controlling bed bugs. Highly insoluble, yeah, log P 6.6. Stable, pretty stable. Highly absorptive, it sticks to glass, it sticks to plastic. And just to compound, because if you look at the structure, there are multiple chiral centers in cypermethrin, and it actually exists as 8 different isomers. Now, the problem with that is that theoretically, your concentration of any one of those is perhaps more than an eighth, but around about an eighth of the total. So that automatically pushes down the limit of quantification nearly by a factor of 10. Historically, the methods of analysis used GCMS with chemical ionization. CI is a good technique. It would be unfair to say otherwise. It's very sensitive, it's very selective. It's quite unstable. It's not an easy technique to employ. And in some cases, it has the habit of covering up important things that are going on in the background, which may bring into question the validity of some of those tests. Now, a better way is GCMS-MS. So hyphenated MS technique, hybrid technique, QQQ or MS-MS, they're 2 terms are synonymous. Introduction of an isotopically labeled internal standard. Our preference is D6 trans-cypermethrin, so 6 of the protons are replaced with deuteriums, gives you a difference in mass. They're separable by mass spectrometry, and by adding that, it allows you to have a fixed marker against which you can quantify. The other one, and this is quite critical, is whole sample analysis. So rather than your colleagues providing you with a sample in a bottle, and you take a portion out of that to analyze, and at each stage, some of the cypermethrin is absorbed into the sample container that you were given, and some of it adsorbs into the pipette you've used to subsample it. You analyze the whole sample immediately, and then anything which has adsorbed into the initial pot, you capture. And that gives you much, much more accurate, much more precise, and much more realistic concentrations. So next slide, please. So further on with practical approaches, silanes, siloxanes, and silicones. Most of these hydrolyze in the presence of water. A common example is bathroom silicone sealant. You pump it around the edge of your bathtub with a skeleton gun. Then it hydrolyzes and cross-links, and then it sticks to the side of your bath and remains slightly flexible and stops the water getting out. If it doesn't hydrolyze, it doesn't work. But when you start putting that in lower concentrations into water, a number of things can happen. It aggregates, it forms dimers, trimers, oligomers, polymers, and the concentrations at which these things start to happen are highly compound dependent. They're also dependent on temperature and time of storage. There's a lot of chemistry goes on in this, and having to understand the underlying chemistry at a really quite fundamental level, and engaging with the sponsor is the subject matter expert, and often they have a wealth of information about how these materials behave. I mean, they've designed them to behave in certain ways, and their involvement is invaluable. Finally, when you actually start to look at the aggregates, at whether it's dimer or trimer or tetramer or something longer, then the simplest, best technique is full scan high resolution mass spec. Traditional mass spec techniques, the MSMS, QQQ that we've alluded to, very, very good for target compound analysis. When you're looking for unknowns, not so much. But the full scan high res, so time of flight is my go-to. That allows you to very, very quickly capture, unravel, and understand what's happening in this complex solution chemistry. Detergents, surfactants, and dispersants is a big, big area. You've got ubiquity because people are washing their hands, but beyond that, the chemistry of these things is extremely complex. You're covering everything from hand soap through to the materials that go into engine oil and sweep away sludge and so on in high temperature engine environments. And these things are formulated to very careful degrees to have the correct useful properties and structures. And they also exist often as very complex mixtures, UVCBs. And the best technique, again, for unraveling what's in UVCBs that I've found is the high res mass spec, the time of flight. Much, much easier to understand what you've got and what's going on than using triple quadrupole. The final point I've alluded to before, but when we were working on some soap materials, you've got a pinch point between the good advice to wash regularly, frequently, and introducing levels of background contamination. And you're having quite difficult conversations with staff members about, can you think about not washing your hair in case you introduce materials into the samples when you come in tomorrow morning? Or would it be okay if you kept the same set of clothes on you were wearing today so that the laundry detergent doesn't contaminate? And they're not scientifically difficult conversations, but on a personal basis, that really can be quite difficult. So can we move on to the next slide, please? The essential toolbox for tackling difficult to test substances. These are the things that you basically need to even start to do this kind of work. UHPLC, ultra high performance liquid chromatography, small diameter, small particle sizes, enhanced chromatographic efficiency and performance. So you can get a better separation from co-extractives, things that are endogenous to the sample. You can also get faster separations. And if you've got unstable samples, unstable analytes, that's a big help. The triple quadrupole, TQMS, QQQ, whatever you want to call it, usually coupled to a UHPLC. That's the industry standard. If you read guidelines, Sante guideline, for example, most of it's written around the fact that you're going to be using something like triple quad to quantify. But triple quad, LCMS won't do everything. Things like volatile materials, and there's a lot of interest in volatile materials still. You really have to look at GCMS, gas chromatography mass spec, and you can couple that with sampling techniques like headspace, like purge and trap, solid phase micro extraction. And by doing those, you can concentrate up highly volatile samples, which you can't concentrate up any other way. You can't liquid extract something and evaporate it down if it's volatile. But those techniques allow you to reach challenging limits of quantitation on those volatile materials. So next slide, please. Hybrid mass spectrometry. So combining two or more mass separation techniques. So triple quadrupole is one, but often it's combining a quadrupole on the front of the instrument with something else on the other end, an ion trap, an Orbitrap, or time of flight. Made no bones about the fact that I'm a time of flight enthusiast. These techniques give you higher mass resolution, and the higher your mass resolution, the higher your selectivity is in complicated samples or for complicated substances. There are trade-offs. Instrumentation tends to be more expensive. It's not so easy to use, but selectivity is key. If you don't achieve selectivity, then your data are not reliable. Over time, these techniques have all become increasingly robust, and there have been gains in sensitivity. So improves the way you can quantify samples. Now, the next one's important, especially for UVCBs. If you've got a UVCB, complex mixture, multiple components, coming back to something like some laundry detergents, you might have 40 different components in there. You want to be able to measure a good number of those in order to be able to turn to the regulator and say, what we've analyzed is representative of the whole substance. The high scan rate is important so you can get enough scans across your analytical peak for the peak measurement to be reliable, but also spread far enough across enough compounds. And that selectivity and scan rate becomes even more powerful when you can combine it with useful simple extraction techniques like Ketchers. So next slide, please. So this is an example of hybrid mass spec. The left side is triple quadrupole, and we've picked out our mass for our compound of interest, and you can see the scale's not very big, but we're from something like 332.5 Daltons to 334. So you've got about 1.5 atomic mass units. Now, when you start assembling the masses of complex substances, there's quite a lot of ways you can come up with a mass which is 333 plus or minus about 0.75 mass units. And that gives you the high risk of having something sat under there which is contributing but isn't actually your test substance. You come to the same substance on the trace on the right, and the span on there is tiny. It's probably about 1,000th of what you have on the triple quad. And that gives you much, much higher specificity and selectivity. We've been using TOF quantitatively at Smithers for 10 years, and we're pretty sold on it. So next slide, please. So some advanced solutions, things which are coming down the pike or are currently available. The DEA-based phases improve retention and selectivity for highly polar analytes. You don't need to use complex mobile phase additives so much. Where Sid was alluding to trifluoroacetic acid, obviously that's potentially going to become even more important. A traditional or semi-traditional approach to highly polar analytes for HPLC was HILIC, hydrophilic lipophilic interaction chromatography, and HILIC is not well liked by most analytical chemists. It's tricky, it's unstable, it takes a great deal of fussing and coddling to get it through, and the DEA phases offer an option to do the things which HILIC does, but with rather less difficulty. Ion mobility, where you can separate ions by mobility in the gas phase. This has great potential, particularly for things like stereoisomer separation. There are separations that you can do by ion mobility which you really can't do any other way. So that offers some really useful separation and selectivity. 2D chromatography has been around for a long time. I was first doing it back in the 1990s. It's remained niche, but it's very useful, where you can separate and isolate peaks on one column, then divert it onto an alternative column with a different phase. So, for example, you can separate an analyte which has multiple stereoisomers, separate out the analyte of choice, then switch that peak directly onto a chiral column, which may not have the selectivity to separate your analyte of choice from all the other materials, and then do the chiral separation in a single run. Tends to make for long run times, but it's a very, very powerful technique to solve those kinds of problems. Nanoelectrospray, step change from conventional electrospray. By improving the what we call the transmission, the amount of sample that goes through into the mass spectrometer, then you get potential for higher sensitivity, lower LOQs, and reducing the matrix effect, especially for macromolecules. Supercritical fluid chromatography, again, if you go back to the 1990s, using CO2 as a mobile phase in a kind of more like a gas chromatographic approach. Now, increasingly, it's using supercritical liquid CO2 as a modifier for what is effectively a form of HPLC, and it offers a completely different selectivity to reverse phase or normal phase, and brings a great many more columns into play than you would normally have for reverse phase separations. There's a quite high entry price in terms of equipment, but it really, really does offer some openings which I think you wouldn't get any other way. So next slide, please. Staying one step ahead. So this is where we see things are heading and where we need to be ready to jump or are already running to make sure that we're in place when these happen. The increasing use of biomaterials. Regulators are keen to have environmentally friendly materials. I think that's not just regulators. I think most people would be happier with more environmentally friendly materials being used. But from an analytical perspective, many of those are more difficult to analyze: large molecules, unstable molecules, variable compositions. And as Sid has alluded to, there are different techniques that are required. Things like RNA analysis, electrophoresis. We're lucky that we have expertise in-house in working with things like RNA. Industrial chemicals. Historically, many industrial chemicals have benefited from, if you like, a light regulatory touch. That, I think, is unlikely to continue as other things have become more tightly regulated. The regulatory bodies now have an attention span to turn towards these, and there's increasing attention on things which may be things like endocrine disruptors. There's new REACH guidance in the system on polymers, and that's likely to have an impact on how they are manufactured and used. We will see increasing scrutiny on nanomaterials. The last point is slightly tangential to OECD 23, but I think it's valid. As we push towards in vitro techniques to replace some of the higher tier tests, so a good example, there's currently a candidate test to look at replacing fish bioconcentration with an in vitro method. Again, nobody's going to be very unhappy about replacing a live animal vertebrate test with an in vitro technique. but the reliability, the validity of those techniques is critically dependent on high quality analytical data to underpin them. And something that we're seeing increasingly is a regulatory push for chemical analysis on terrestrial testing. That really is something that we're seeing ever more of. So to the next slide, please. So these are our conclusions, that we regulators and the general public are expecting novel chemicals to be environmentally friendly, and that this is a positive development. But those environmental properties, as Sid explained, bring different analytical challenges. Things that regulators have ignored or only lightly touched on in the past are receiving significant scrutiny, and that seems unlikely to change. The importance of up-to-date and accurate physical chemistry data, you can't overlook that. Nowadays, the simple less than, greater than values that one finds, those will no longer be acceptable. And that there's a diverse analytical toolbox is needed, is absolutely essential to meeting those challenges of the difficult to test substances. So with that, I'm going to hand back to Phil, and thank you all for listening.
Okay, thank you, Mitch. Yeah, so that concludes the content of our webinar today. So at this point, I'd like to open the floor to the audience for a bit of Q&A. So please, if you could use the Q&A box, please send over some questions and we will do our best to answer them live. So a couple of minutes to receive some questions. Okay, a couple coming through here. Okay, right. Sid, do you fancy taking this one? Are toxic chemicals deemed to be difficult to test substances?
So, well, in terms of handling, that would certainly the answer would certainly be yes. But that's not really what we're here about. So I would say in terms of chemical analysis, the answer is it depends. If I could give some examples. So paraquat is, I think, a good example of a test substance which is very toxic. It's a herbicide. It's a broad spectrum herbicide, but it's also known to be quite toxic to mammals. And it's also, to an analytical chemist, it's also notoriously difficult to analyze. And so that's an example of something that, yeah, is toxic and difficult. Sort of on the other end, I'm trying to think. So if you've got something that's toxic, but not especially difficult to analyze, I think an example would be fentanyl. Is an opiate, as I'm sure most of us have heard about. It's even at low concentrations is very toxic to humans and probably most mammals, I would assume, but it's not especially challenging to analyze, and of course. A lot of it's dependent on dosage. It really is, what do you define as toxic? So the answer really is it depends on. There's lots of factors. And there are some toxic substances that are difficult and then some that aren't. Yeah, it entirely depends.
Okay, thanks, Mitch. Okay, there's another one here that I've picked out of the list. Mitch, is there a chemical property that poses more challenges than others?
Yeah, to my mind, the single most challenging property is ubiquity. It's dealing with those substances where you've got backgrounds, plasticizers, silicones. I mean, historically, metals analysis was always a problem because it's very, very difficult to get away from background levels of lead, aluminium, iron. But that's a real challenge, because also what you find is that For something which is highly insoluble, but is also ubiquitous, the difference between the lowest dosing concentrations that an ecotoxicologist is trying to achieve and the existing background is very, very small. If you go to something like the SANTE 12830 guideline, what you're looking for there is that your limit of quantification is basically 3 times higher than what you find in your unfortified control sample. So as that unfortified control sample gets higher, the place you can put your LOQ has to go higher with it. And it's always that fight to reduce that background in your exposure media, in your reagents, in your consumable supplies, so that the impact of the endogenous is reduced compared to the treatment dose that you're actually trying to investigate.
Okay, thanks, Rich. And Sid, again, the same question to you, really, based on your background and what you've been involved in. What do you believe is the property that poses the most challenges?
I guess it overlaps a bit with ubiquity as persistence and extremely stable compounds. Historically, it poses a challenge, at least in your test system design. I guess if we go back to cypermethrin, it's not especially ubiquitous in the environment, but it is quite persistent and has absorbing properties, and it's stable. And so yeah, I would say something that's stable can also in its own way cause challenges.
Okay, thanks, Sid. Okay, there's probably time for one more. I'll just pick this one out of the list. Oh, okay, Mitch. Well, we were discussing this last week, but will the proposed changes to REACH and the requirement for polymer testing pose any challenges?
Yeah, I think it would be unreasonable and unfair to say they won't. Polymer chemistry is something I've been peripherally involved in throughout my career, and polymer chemistry is a discipline in itself. Understanding how polymers work and the properties of them is a different field. There are techniques which you're not commonly going to use for most of the conventional small molecule analytical techniques, things like size exclusion chromatography, to establish your mass boundaries and the extent to which mass varies within the sample. That's not a difficult technique, but it's one that you'd need to get to grips with. Often those, particularly the very large polymers, they're not very amenable to mass spectrometric analysis. So you need something else as an end determination. Evaporative light scattering detectors work. There are different challenges, but analytical chemistry is all about challenges, and we've been meeting challenges since James Lovelock developed the ECD to understand the organochlorine pesticide problem. So we'll keep on finding ways to understand things.
Okay, there's a number of further questions in the chat, but I'll leave it there for now.